Good evening, ma'am.
Good evening, sir. You can
Good
continue,
evening.
sir.
Yes, ma'am. Thank you, ma'am. Good evening, my dear students. Once again, you are all are welcome to the biological session. The continuity of the food strategy. Okay, my dear. Last class, what we have discussed already, we know about. See, interspecific hybridization. And also, another one is called as what is that, crossbreeding, outcrossing, all those things already we discussed, isn't it? Today, we are going to discuss about the remaining part of the, which one, this lesson. First of all, is it no? A control breeding experiment. Okay. Control breeding experiment. So what are the two types of control breeding experiment already you know about? One is called as artificial insemination, simply called as AI. Okay, media. So this is very, very important. Most of the cases, most of the question paper, it is present. What is by artificial? Artificial means you know about we are collecting the spam and spam particularly. The condition is called as artificial insemination. Collection of spam from the animals. Okay, animals. Okay, that condition is called as what is that, artificial insemination. And also one more is that it is also very, very important. Multiple ovulation and embryo transfer technology, simply called as MOET. That is very, very important in NEET point of view, my dear. So many times they ask the questions. So many times. Okay, what is the expansion of MOET? Multiple ovulation. Ovulation means nothing but egg. Okay, here what happened, the artificial insemination means spam. Don't forget these two things, my dear, because both are very, very important for the breeding, controlling of the breeding. That is very, very, very important, my dear. So here the spelling mistakes there, breeding. Okay, breeding. Don't forget. See one by one, my dear. First of all, artificial insemination. What is meant by artificial insemination? How it will be collected? How it will be stored? How it will be utilized? These are the 4 points is very, very important, my dear students. First of all, artificial insemination is nothing but the semen is collected from the male. Already you know about the sperm collected from the male that is chosen as a parent and injected into the selected female genetical organ. So this is the point. Here, what is the meaning of artificial insemination is nothing but the collection of sperm. Okay. Already you know about collection of sperm. There is a method is the male from the male. And then after the collection, this can be transferred from the one place to another place, one country to another country, one district to another districts. Okay. And also after the collection, it may be stored and then it may be transferred into the respective selected female genetical tract. Okay. That is very, very important. Just like a test tube baby. Okay, my dear. So here what they happen here, the surrogate mother as well as the sperms also, egg and sperms also collection, donor, then uniter and also undergoes a meiotic cell division already in our board. After that, it will be introduced into the female genetical tract. Already you know a little bit. That is called as what is that, implantation. Okay, my dear. So that is called as what is that? Implantation. But here we are collecting the sperm only. That is the purpose. Okay, purpose. Then what happened here? Just see it. This is the first point. And second, when a bull inseminates a cow naturally, approximately 5 to 10 billion sperm are deposited in the female reproductive part. Normally, okay. How much sperms they ejaculated or ejected at the time of coitus? 5 to 10 billions. Don't forget, my dear. After that, once this is normal, okay, these two are interconnected. These two are interconnected. The semen may be used immediately or can be frozen. Frozen means, you know very well about under the low temperature. So many times they were asked the question, my dear students. Don't forget this one. Not only this one. Frozen by cryopreservation. What is the meaning of cryopreservation? Cryopreservation is nothing but the one of the technique they mostly used for the collected specimens such as sperm, okay, and egg, and spores, and seeds, okay, and also some of the microorganism also. These are all under kept a particular low temperature with the help of liquid nitrogen at minus 196 degrees Celsius. This is very, very important, my dear student. 3 to 4 times they ask the question. Just go and check it, the question paper. You get the ideas. The semen may be used immediately or can be. Immediate means what happened? Here we are collecting, isn't it? We are collecting the sperm. Directly, it can be transferred to the 1 or 2 days immediately. Or immediately it will be transferred to the, what is that? Superior female. Suppose it will take some more times. You have to travel for some times, you know, that time what they are doing, the scientists, they are doing the cryopreservation, otherwise called as frozen technology. Technique. During this process, which liquid is used? That is very, very important. Liquid nitrogen. Okay, liquid nitrogen at 196 degrees Celsius. Don't forget, my dear student, this is also very, very important. Just to go and check it to 2017, 2018, 2019. All the question paper, the question is there, which is used as a source of gene bank. Why? Because they store in the gene bank. Please. Okay, my dear. This is very, very here. The 3 point is very, very important. The artificial insemination is nothing but the collection of sperm from the male. The second one is, what is it, you know? It can be stored where in the cryopreservation technology or technique with the help of chemical substances is called as liquid nitrogen at which temperature? minus 196 degrees Celsius. Mostly they were used for the gene bank. Okay, my dear, this is very, very, very important. Okay. The artificial inseminations helps overcome several problems or normal matting. Okay. Okay, my dear, this is, what is that? See that artificial insemination help overcome, okay, normal matting, normal reproductive process. What are the difficulties they are having? It can be rectified by this method. So this is also one of the NEET question, my dear. And second one is by this process, desirable character. That is very, very important. Desirable characteristics. For example, already we discussed. What is that? 2 things is that. For example, suppose take an examples of cow or buffalo, lactation. Lactation is, what is the meaning of lactation? Lactation is nothing but the secretion of, what is that? Milk, my dear, milk. Okay, and also high yield of progeny, and also disease resistant capacity. These are the desirable character, my dear. Don't forget. Desirable character. When you are selecting the superficial character of the male, so you have to consider these are the parameters. Okay, my dear. Or combined in the progeny. So this is very, very important. Main desirable character, what are the high quality of milk, and that the young one, the offspring, are to superior quality, highly resistant capacity, and survival fitness also. That is very, very important. Okay. The artificial insemination is carried out when the mating of mature male and female animal is low. Okay, artificial. The age is different. Okay, here take an examples of 15 means here 13 years. Okay. To improve chances of the successful production of hybrid, that is also very, very important. Already told you, isn't it? Successful hybrid, that is called high yield progeny. Other means are also used. See 3 here. The artificial insemination help overcome the several problems: disease resistance, milking, survival fitness, superior quality, and also other things also. Even taken examples of muscles, okay, and also hard work properties. All those things is there. And desirable character. Artificial insemination is carried out when the mating of the mature male, then only, okay, and the female animals of mature male and female, okay, don't forget. Animal is low. To improve chances of successful production of hybrid, this is also very, very important, already discussed. Okay, my dear. Okay, see here, more than, listen carefully, this is very, very important, my dear students. More than 6,000 artificial insemination centers have been established in different parts of the country to benefit the dairy and farmer at village level. Why they are using? See that village level. Okay. One such center is Indian Veterinary Research Institute. This is very, very important, my dears. This is very, very important. Okay. So many times they may ask the question IVRI. IVRI. Okay. See that? India only. Indian Veterinary Research Institute, Izzat Nagar, Roy Berli, Uttar Pradesh. Izzat Nagar, where is it, you know? Uttar Pradesh. Okay, UP, simply called as what is that, you know? UP. Don't forget, my dear student. This is very, very important. So many times they were asked the question, IVRI, where it located. First of all, they may ask the question, expansion of the IVRI. Okay, expansion. This is the first question, expansion. Expand. Which one is the correct expansion of the IVRI? Expandable manner. And second point is where it located. Okay. Located. Izzat Nagar, Roy Berli, Uttar Pradesh. Which state? State is also one question. And IVRI. And 3 question is in the single paragraph, my dear. Don't forget, my dear. Okay. So go to the next one. So what are the advantages of the artificial insemination? Sometimes they may ask the question. Main advantage already we discussed, isn't it, my dear? What are the main advantages? How it will be? See here. Artificial insemination has a number of advantages. What are the number? Many cows up to 3,000, maximum 3,000 number of cows can be fertilized by the semen collected from one bull. Don't forget. So one male, okay, one male, superior, okay, male, they can fertilize how many cows? 3,000 cows. 1:3,000. Imagine 1:3,000. Imagine. So this is the first point. Superior quality, we can transfer any cows, those who are having the mature ability, we can easily transfer to the female reproductive part. As a result, superior quality of the offspring we will obtain. That is very, very important. And second point, frozen semen. Just note it down. Frozen semen means minus 196 degree Celsius liquid. Okay, nitrogen. Okay, my dear, liquid nitrogen. Liquid nitrogen. The frozen cement can be stored for a long period and transport to the remote part of the country. That is very, very important. Why we are storing this one, Okay, then only it can be transferred from the one place to another place, one country to another country, one state to another state, and also in village also. State to village, village to state, whatever it may be. So this is the very, very important point. Why we are preserving, cryopreservation. And third one is this method is economically and what is gives a high rate of, this one is also wrong. It gives, okay, gives high rate of successful fertilization. When you are this type of artificial insemination, it will be succeed how much, suppose one bull, they can fertilize 3,000, isn't it, my dear? 3,000 cows, isn't it? So how much is possible, maximum percentage, 2,980, 2,990, maximum percentage. That's why it's high rate of fertilization is possible. There is no infertility capacity. That's why, that is the reason. That's why we are selecting the male cow. Okay, bull, sorry. Okay, we are selecting the male one. It has a highly superior, that is the quality, superior quality. Okay, my dear. Go to the next one. And second methodology is called as, what is that, here what they did, first one, a sperm collection. Here what they are doing, second one is called as multiple ovulation and embryo transfer. Embryo transfer. What is the meaning of embryo? Already we discussed, isn't it, my dear? The technology is called as NEET. Don't forget, NEET is nothing but the very, very important NEET question. Okay, just go and check it. Cross matches. Okay, matches, and also expands, and also direct questions. These are the very, very important. And also NEET is used for the herd, a group of animal improvement. What is the use of this one? The group of animal improvement. In this method, cow is treated with the hormone. See here, cow is treated with the hormone such follicle stimulating hormone. Which one, follicle stimulating hormones? Sometimes they ask the question. In NEET, which hormones used for, what is that? Method of NEET, like that. Follicle stimulating hormone, FSH. Simply. To induce the follicular maturation and super ovulation. What is the meaning of ovulation? Super ovulation means when you are using this hormone. So, for example, take an example. Cow is there. Okay. Cow is there. See that. Cow is there. When you are introducing the FSH. Okay. When you are introducing the FSH, the cow, what happened? They will attain maturation fastly. Attained is that maturation. Okay, madam. Maturation. What maturation? Ovulation maturation. Ovule. So otherwise called as egg maturation. So we will get short period. How many is that? When you are introducing the FSH, automatically cow also attain the maturation. Which one is the oval maturation takes place? So you will get the egg very short periodly. Okay, normal. Take an example. Each and every in human beings, just take an example of 25 days. You have to wait for the single egg. Single egg. Okay, maximum 20 days. But in this category, just imagine, I'm not sure, but imagine for 30 days like that, 30 days or 40 days, like just imagine 40 days means per years, how many days we will get it? 6 to 7 times. Okay, my dear, that is the reason. See that the which leads to the formation of 6 to 8 eggs per cycle. See here, 6 to 8 per cycle. How many egg we will get it? 6 to 8 eggs rather than 1 egg per cycle. Normally, which one the cow yield only 1 egg only per cycle. 1 egg, just like a human being, 1 egg per cycle. Suppose when you are introducing FS, guys, how much you will get it? 6 to 8. See that ratio, 6 to 8. Okay, my dear. So next, this is very, very important. Don't forget, my dear. First, follicle stimulating hormone. So as a result, what happens? The egg maturation is also takes place very fastly, and not only that fastly, and also number is also many-fold. Next, the cow is either matted with the superior bull or it may be artificial inseminated. This is normal one. Normally, either matted, normal, other with the superior bull, or it may be artificial inseminated. When you're what is that? Normal mated means what happened? You will get the only one, which one progeny. Suppose artificial means we can introduce. So here the 6 to 8, what is that? Egg produces, isn't it, per cycles. So here when you are doing this one artificial insemination, how many cows you will get it? 8 to 6 progeny will get it. That is very, very important. See here, my dear students. For example, see here, a cow is when you are introducing the FSH follicle stimulating, isn't it? Follicle stimulating hormone, isn't it? Introducing means normally the cow yield 1 egg per cycle. This is the normal one. When you are entering FSH, what happen? 6 to 8 egg per cycle, isn't it? They will give cycle. In this category, after the injection of FSH, 6 to 8 egg you will obtain. So this time artificial insemination takes place, So sperm is also reached. How much is the already discussed? So the progeny, it will be how much you will get, 6 to 8 offspring is possible. Yes, what is that? Possible. Isn't it, my dear? That is very, very important. So this point, don't forget, my dear students. First of all, you should know which hormone is responsible, how many egg you will get it, artificial immunization, how it will be possible. See that one. Then, next. The fertilized egg at 8 to 32 cell stage. What is that? This is called as blastula, gastrula. Already we discussed it. When the fertilized egg undergo the cell division process, which is cell division, meiotic cell division or mitotic? Mitotic, isn't it, my dear? Mitotic cell division. When it attains 8 to 32 cell stage or isolated and non-surgically and transport to the surrogate mother, the female carrier fetus of the other animals. Okay, my dear, this is very, very important. This is also, it is also one of the methodology. Okay, the collection of sperm and also inserted sperm and inserted to the, which one, A animals, isn't it? Animals. After the cell division started up to 8 to 32 cell stage. Okay, cell stage. Isolate or collect the 32 cell stage egg. Okay, collect and also inserted, a transfer non-surgically. Non-surgically means what is it? It's just similar to the test tube baby to the surrogate mother. Okay, surrogate mother. Okay, my dear. So this is very, very important. So next one is the genetic mother is available for another round of super ovulation. Genetic mother is also available for the super ovulation. Super ovulation, what is the meaning? 6 to 8 egg per cycle. Okay, and then next, this technology has been widely used for the cattle, sheep, rabbit, buffalo, mare, etc. Okay, because take an example. The white, for example, only I told you. Okay, I am telling. White lion is there. Okay, only one white lion is there. Superior. Okay, it collect the sperm artificially. It may be inserted into the superior. Okay, normal superior, but not female lion. Okay, after the 32 cell stage, it collect and also transfer to the, which one you know? Surrogate lion. Okay, now we will get it. How much you know? 6 to 8 offspring of white tiger, or sorry, white lion. Isn't it, my dear? This is very, very important. Just like that, we can do it any other animals. Take an examples of sheep, rabbit, buffalo, mare, etc. respectively. And another one, by this process, less meat. And also one more is there. Less meat means what is that? Some of the animals contain more fat. Okay. But here, when you are using this type of technology, what happened? Less meat. Lean meat process. Lean meat means the muscle is there, but low fat is there. That is very, very, we can get it. Suppose take an example, is rabbit or sheep. It is a lean meat as well as a low lipid means what happened? It will not affect any human body. See, the same thing. Yielding bulls and milk yielding cows are developed, which is very helpful to increase the herd size in the short time. This is also very, very much. Within short period, we'll get number of progeny or offspring. That is very, very important, my dear student. In this part, don't forget here, very, very important. Artificial insemination is very, very helpful to the human society. Okay, my dear. So go to the next one. So that's all about the, what is that? Artificial insemination, moital. Next, we'll pass on to the next one is the beekeeping. What is the meaning of bee? Honey bee. Okay. Okay, my dears, honey bee. What is the uses of honey bee? The honey bee collecting the nectar. Honey bees. Okay, it's a insects. The science study of the insect is called as entomology. The science that study of the insect is called as insect. Or called, what is that? Entomology. Okay, don't forget, my dear. Sometimes they may ask the questions. Okay, suppose we are rearing the culture of the honeybee is called as apiculture. The apiculture is the maintenance of the hives of honeybees for the production of honey. Okay, this is very, very important, my dear. Apiculture is nothing but one of the process, just like agriculture, arboriculture, silviculture, oyster culture, like that. Apiculture is also one of the process. But here, what happened? You know, they are, what is that? Rearing the honeybees, maintaining of hives of honeybees for production of the honey. There are several species of the honeybees that can be reared, or these, the most common species in India, Apis indica. See here, there are 4 types is there. Apis indica. Indica is a normal bees. Apis mellifera. Apis mellifera. Apis dorsata. Okay. So, Apis dorsata. These are the three types of animal is there. Okay. The Apis indica is the normal size in nature. They are all depends upon the three things is there. Depends upon their properties. This depends upon their structure. First of all, size. Size is also vary. Okay. And then second one is, what is that, production of honey. Production of honey. And also the weight. Okay. The Apis dorsata is one of the important. Okay. This can be, the hives may be, how many kilos, 25 to 30 kg of honey. 20 to 30 kg of honey. The Apis indica maximum 2 to 3 liters. Apis mellifera only 400 milligram, 500 milligram or 1 kg below. These are the types. Okay, my dear. So these are the important. When you are going to study that, beneficial effect. At that time we will study this one. Okay. See this one. This is called as Apis indica. See that it collecting the nectars, honey from the flowers. After that, it may be changed in some other forms. And also store in where, hives. Okay, the size is normal. It has a sting ring. The sting, it contains one of the chemical substances called as formic acid. What is that? Formic. F-O-R. Formic. Okay. Formic acid. That's why you are getting pain. Okay. Not only this, and also some of the insect, just like this. See the next one. The economically important of the honey bees. Just imagine honey. First of all, honey. Okay, it is a rich source of the fructose. Don't forget, my dear students. The honey is contained which sucrose is very rich. Sugar molecule is very high content. At the same time, which one is a high content? Honey is made up of what are the types of sugar molecule? They may ask the question. First of all, is note it down: fructose and water and glucose. These are the two sugar molecules present in it. Okay. Minerals and vitamins. Commonly, fructose, water, and glucose, and minerals and vitamins also present in it. Honey is a sugary shrub. Liquidity. It's a semi-solid. It's a colloidal, semi-colloidal nature. High density compared to the high density. Okay, my dear, high density. And second one is, note it down here, the sugar molecules and fructose and minerals and vitamins. So the pH is near to the base, 6.8 pH. The water, 7 pH, isn't it? So it is near, only 0.2, only less. See how much amount of water molecule present in it in this category. Depends upon the, some of them are liquid in nature. So the high content of the water molecule, 75 to 25%. Reverse manner they told. 75 to 25% of the water molecule. It may like this. It may be very depends upon their honeybees. How many percentage of the minerals present? 3 to 3.3% of minerals. Percentage. 3.3%. Percentage denoted as 100. 100 ml of honey contain 3.3 milligram, like that. Okay. Abundance vitamins, already in our board. Vitamins, what are the vitamins present in it? So many things is there. Okay. Fat soluble vitamins, particularly vitamin A is more abundant. Vitamins A. Okay. And also D. Okay. E also there. K is very less. K is not there, like thing. These three things is mostly present in it. See that L-fructose, the fructose is also L-fructose, that is also levulose, 41%, glucose, 35%, Sucrose 1.9%. Which one is the highest concentration of the sugar molecule present in it? Means you have to tell which one is the correct answer. Fructose is high content. Don't forget, my dear. This is also very, very important. So many times they are asked the questions. F is fructose is 41%. G is 35%. S is how much is there? 1.9%. So fructose is the high content. It is also asked so many times. NEET. The honey contain which carbohydrate is mostly highly present. High content of the carbohydrate. Okay. And one more is it can be used as a tonic, ligative, and sweetening agent, and also act as a good antiseptic. When you are applying in the wound, what happens? It acts as an antiseptic agent. Okay, my dear. Don't forget these things also. And the next one is bee wax. Don't forget bee wax. It is the product of the industrial importance. Normally, the bee wax is also used for the manufacturing of the so many cosmetic savings cream, face cream, ointment, plaster, pencil, electrical goods, toothpaste, and lotion, protective coating, ink, paints, polish of various kinds of carbon paper, and candles. Okay, mate, don't forget. These are the important of the bee waxes. It is also used to model and modeling, making, and printing industries. The model means, the clay model like that. Here, the wax model is there. Molding. Okay. Molding is also there. Making, printing industries. These are the important things. See here. Manufacturing of cosmetic savings. Cream, face cream, ointments, plasters, pencil, electric goods, toothpaste, lotion, protective coating, ink, paints, polishes of various kinds of carbon paper. Okay. And also the candle. These are the most important things. Okay. Go to the next one. It is also used to the laboratory for microtomy. What is the meaning of microtomy? Microtomy is nothing but the instrument used for the, which one histology. What is my histology, my dear students? Histology is nothing but the study of tissues. It's called as histology. Here, what they do the microtomy. What is the meaning of micro? Micro means minimum, isn't it? Tomy is nothing but the instrument. It's an instrument, just like a cutting machines. Okay, cutting machine is there. It is used for the cancer cell section process. Okay, suppose for example, take an examples of any one of the cell. Take an example. Cell look like this one, isn't it, mate? This is considered a cell, isn't it? Normal. Suppose if it is modified due to the unfavorable conditions, carcinogenic agents, ultraviolet rays, physical and chemical, biological changes which occurs within the nucleus, means what happens? The cells also multiply into different in nature. Multiply. This is cancer cells after the multiplication. Probably identified cancer cell. Normal to cancer cell. How will you identified visually means they were used, which one of the instruments is called as microtomy. It is similar to that of the cutting machines, just like a papers. Okay, you have to take the sample from the person. See here. See here, this is the normal cell, isn't it? Just imagine. This one is considered a normal cell. Already we draw it in the previous slides. Okay, normal one. It's also converted to cancer, isn't it? or tumor, isn't it? Tumor. Okay, this one is considered as a normal. This one is considered as a cancer cell. How will you identify? How will you identify these things? You know, so examine under the microscope. You will you will see this type of structure. Means what is the meaning? Before that, you have to collect the tissues. For example, this is considered a part tumor tissues. Have to take the cross section. You have to take the cross section. After that, undergo the staining process. And then examine under the microscope. Examine under the microscope. So this in this part, why we are taking this? No, they say after the collection of these samples, also placed into the wax block like this. Okay, just imagine this is consists a wax block. Here the tissues is placed here in this place. Okay. This is consists a wax block. Okay. The microtomy is nothing but the instruments. Microtomy is the instruments. It has a blade like this. What happened? It cut like this. Okay. So it has a blade like this. Okay. It cut like this. Like this means you will get the first the block like this. Thin block you will obtain. Here the cell also cut into the small piece. Like this. So after cutting, the machine is like this. Machine is like this. Microtome machine is also there. Okay. You have to place the block here. The machine is cut here. Okay. Here. You made a cut. So this is the block. Okay. Block. So what happened? No, the machine is like this, up and down. They can move from up and down. So that time what happened? You will get the slice like this. After that, you have to undergo the staining process. Then examine under the microscope. You can see the structure like this. You can see the structure like this. So we come to the conclusion. This is the normal cell. This is the affected cells. Okay. Then only we can see the differences. So that's why what I told here, you know, so two purposes. What is my microtome? Microtome is nothing but the instrument used for the cancer tissue slicing process. And one more is there, why we are using this one, you know, the cells also after collected may be placed inside the wax box. Okay, wax block. That wax block is also, the wax is also obtained from where, you know, honeybee. That is the main. Okay, my dears, that is very, very important. Okay, go to the next one. Okay, that is very, very much. See here, the cancer cell section with the common was the block preparation of the tissues. Very, very important, my dear student. Microtome, mostly they ask the question NEET. Once upon a time, they asked the question. It is secreted by the abdominal wax gland. So how the waxes are secreted means honeybee. Okay, honeybee abdominal wax gland is there. Honeybee secreted the abdominal wax gland of the worker bees. What is the meaning of worker bees? Maximum worker bees. So think that it is also NEET questions. Which one it has a which one, you know, wax gland, like that they may ask the questions. Okay, worker means maximum sterile female, don't forget. Okay, it possesses the hardening substances from the cephalic gland. The name of the gland is called as cephalic gland and resin called as propolis. These two is very, very important. Right? Two is very, very important. Cephalic gland. cephalic, resin, gum-like substances called as propolis. Forms pollen grains. Already told you why it is the waxes are look like a yellow in color, orange, yellowish orange in color, mean due to the deposition of the pollen grain. Pollen grain obtained from where, the flower pot of the anther. Don't forget, my dear students. Okay, so many times they were asked the question, why it look like a yellow color or orange color? Because the deposition of the pollen grain, it contain carotenoid. Pollen grain is made up of which one, carotenoid. That's why there is a sticky substance. Is that sticky substance also collected from the nectar gland? Here you must note it on 4 to 5 point is there. First of all, microtomy, laboratory used for the cancer cell sections. First point. Second one is the wax gland, abdominal wax gland. That wax gland is called as what is that? Cephalic gland. Okay, cephalic gland. Mostly that cephalic gland present in the worker bees. And the worker bees, which one, sterile female. Okay. And also the resins, both are present in it. Cephalic gland and resins also called propolis. The propolis obtained from where, pollen grain. The pollen grain where it is present already in our board, plant. Which one, pollen grains where it's present in the flower, isn't it, my dear? Flower. Okay. Here the anther is the pollen grains. The pollen grain where it is inside the anther. Okay, my dear. So that's why after the collection of pollen grains, what happened? Once the dehiscence is happened, the anther is like this. Dehiscence is happened. Okay, the pollen grain comes out. When the insects, okay, adhere on the surface of the pollen grains, what happened? It adhere on the leg also, in the leg also. And also it's also transferred to the one flower to another flower, automatically it be move on. Okay. It may be the pollination possible. That is called as cross pollination. Already we discussed. Isn't it, my dear? Okay, what is the next one? See then propolis. See that propolis is used to treatment of the inflammation and superficial burns. Maximum is just like a waxes. Okay, waxes or oil-like substance, resins, isn't it? And another one is called as bee venom. It is also one of the poisonous ones. See that it is also extracted from the sting of worker bees. Sting worker bees. That is called as formic acid. I told you, formic acid. Okay, it gives paining. It is used to the treatment of rheumatoid arthritis. Arthritis means what is that? Bone. What is that? Pains. Just to take an example, any one of the knee joints or some other things in rheumatic, bone related. That time what happened to the formic acid when you are that means what happened to the honeybee is stinking in the particular places. What happened? No, it is a treatment of the rheumatic arthritis, joint pains. Okay, very, very important. Sometimes they may ask the question, rheumatic arthritis is also cured by the one of the bee venom. Okay, is it correct or not? Like that, it's a true or false. Stinking, where it obtained, it is also worker bees. Worker bees only, what happened? It can bite, sting, their venoms. That is very, very, that's why you are getting the pain. Okay, and another in Chinese people, they are used in this methodology. Bee venom is also used for the treatment of the painkiller. Okay, sometimes. And what about the bee also affected by some diseases due to the microorganisms? See that. Nosema disease. What is the main of first one is Nosema disease. Is caused by the Nosema apis, a sporozoan. Don't forget which one of the sporozoan produce the bee diseases. Nosema apis. Acarine disease. Another one is also acarine disease caused by the Acaripis woodi, a mite. Aspergillosis is caused by Aspergillus, is nothing but the fungi. Okay. And septosemia is caused by Bacillus asepticus. These are the bee diseases. Just note it down, my dear. This is very, very complicated, but note it down. B. N-A-A-S. N-A-A-S. Mnemonics. That is called as what is a Nosmia, Acarin, Aspergillus, Septozemia. These are the corresponding name is coming here. Nosmia, Nosmia herpes. Acarin, Acaripes woody. Aspergillosis, Aspergillus. Septozemia, Bacillus aspiciptasis. Okay, my dear. This is very, very important. Just to go through it, you will get the idea. Sometimes they ask the question in this part. Okay. And next one is called as fish productions. Fish is a cheap source of animals production in the food. Fish production include the finned true fish as well as the shellfish such as the prawns and also mollusks. There are two types of obtained fishes. One is the farm natural resources. Normally, what is that? We are getting the natural fish. What is that? Take an example: sea, river, pond. Okay, my dear. So these are the important, which is called as capture fishing. Now, other way by fish farming, which is called as culture fishery. This is natural. This is artificial. Okay, my dear. The water source of the fish can be either seawater or fresh water, such as river or ponds. Already told you. Okay, my dear. Fishing can be done both by capture and culture fishes. Capture and culture. Okay. So this is also called as pisciculture. The science that deals with the study of the fish and culture of the fish is called as pisciculture. Next one is color of revolution. Just note it down here. So many times they were asked the question in this part, my dear. First, you should note it down. Okay. Which one is green revolution? Green revolution is denoted as the agriculture oriented. Agree. Green. What is that? Agree. White, just imagine white. Which one is white in color? Take an examples. Here, egg is also white, but it will not come to the white color. See, white is also known as milk and dairy. Milk. Take an example, milk. Okay, so white color. Black revolution, petroleum. It is also called as, what is that? Gold revolutions, golden revolutions. Okay. Black revolution, take an example, not only the petroleum, take an examples of coal. Okay. Pink revolution, sometimes they ask the question, pink revolutions, onion and bronze. Don't forget, my dear, this is also asked so many times. Bronze, NEET. Okay, NEET. And brown revolution, leather and cocoa. Leather and cocoa. Blue revolution, fish. Golden revolution, horticulture. Here, agriculture. Here, golden revolution. And also here, the yellow revolution, oil seed. This is very, very important, my dear student. This is also NEET questions. Golden also, they take an example, turmeric. Okay. Blue revolution, just note it down. This is NEET question, my dear. In this part, in this session, don't miss it. Very, very important. Okay. Green revolution means agree. White revolution means milk. Black revolution means what is it? Petroleum. Okay. And then pink revolution means just note in onion, prawns. Brown revolution, leather and also cocoa. Blue revolution, fish. And golden revolution, horticulture. And yellow revolution, oil seeds. Okay, my dear. See here. Green revolution, food grain production. White revolution, milk production. Yellow revolution, oil seed. Blue revolution, fisheries. Golden revolution, fruits. Black and brown revolutions, non-concave venial energy, convenience energy. Silver revolution, note it down. This is also very, very important. Silver revolution is called as egg. Silver revolution is called as, what is that? Egg. Silver revolution. Round evolution, it is called as potato. Okay. Potato. Red revolution, example, take an example, tomato and meat. A gray revolution is nothing but the fertilizer. These are the important points, my dears. These are the important revolution is also there. Just to go through it, you will get the ideas. Okay, media. So this is the important points. So we will see the remaining things on the next class, my dear students. Okay. Today we will finish to up to this level only. So remaining things we will see the next classes, my dear. Okay. So what are the things already we discussed? Just we will summarize all those things. Just to imagine what are the things. First of all, you should note it down. What is meant by artificial insemination? How will you control the breeding experiment? It may be divided into two types. It may be carried out by the two things. One is called as artificial insemination, and second one is called as multiple ovulation, simply called as MOET. Multiple ovulation and embryo transfer. It is very, very important. Okay. The artificial insemination is nothing but the collection of the sperm from the superior male and also transfer to the female genital tract. Once common sperm, it will be ejaculated at the time of coitus, 5 to 10 billion sperms. How it will be stored? It is also used in the form of frozen technology by cryopreservation in liquid nitrogen at minus 196 degrees Celsius, which is used as a source of gene bank. Don't forget. And not normally, what are the advantages here? Artificial insemination helps overcome several problems or normal matting. What are superficial character mean? Desirable characters, that is called as lactation, secretion of milk, and high yield progeny, disease resistance, all those things we will get it. Production of hybrids also. Okay. And also it's also carry out from the one place to another place. Okay. Transport mechanisms. More than 6,000 artificial insemination centers have been established in different parts of the country to benefit the dairy and farmer at village level. One such center is Indian Veterinary Research Institute, IVRI, Izzat Nagar, Roy Berili, Uttar Pradesh. Okay, Uttar Pradesh. Don't forget, my dear student. This is very, very important. Don't forget. Artificial insemination has a number of advantages. Already in our many cows up to 3,000 can be fertilized by the semen collected from the bull. Frozen, already we discussed. Transfer from the one place to another place. That thing's also already discussed. This method is also highly economical and also gives a high rate of successful fertilization also. 1 is to 3,000 cows. Don't imagine. Might is another type of technology. Here, what happened at the egg transformation takes place, or ovulation takes place, transformation takes place. MOET is used for the herd group of animal improvement. So what they had, one of the specialized hormones is also introduced in the cow. At the time, what happened? It may be produced more than 3 to 5 eggs, more than 5 eggs, 6 to 8 eggs also per cycle. Okay, next the cow is either mated with the superior bull or it may be artificial inseminated. Okay, and after that, what happened? It's also collected from the mother, 8 to 38 cell stage, and also transferred to the surrogate mothers. Already in our, but that thing's also already we discussed. Genetically, mother is available for the another round for super ovulations. Likewise, we can, what is widely used for the cattle, sheep, rabbits, buffaloes, and so on. So by this process, lean meat, low lipid yielding bulls and milk yielding cows are also developed, which is very helpful to increase the herd size in the short times. Beekeeping is nothing but the apiculture. Culturing of honeybee is called as apiculture. There are so many types of bees present in the countries worldwide. Apis indica is the mostly present in the Indian bees. Apis indica, Apis dorsata, Apis mellifera. Okay, these are the 3 types have been reported according to the size, and production of honey is also very high. Also, size is also very big in size. Okay, see that Apis indica. Economic importance of the honeybees. Just of all, you note it down. Honey bees produce honey. It is rich source of fructose, water, and glucose, minerals, and vitamins. Honey is a sugary shrubs with the pH of the nearly 6.8, having 75% to 25% of water, 3.3% of the mineral, and abundance of vitamins. The fructose, L-fructose, levorotatory. L is denoted levorotatory. D is denoted dextrorotatory. Levorotatory. L-fructose. Levulose, 41%. It's the highest concentration of the sugar molecule in the honey. Glucose, 35%. 1.9% of sucrose. Dextrin is also present in 1.5%. It is also act as a good antiseptic agents, sweetening. Beeswax is also prepared. Beeswax is also used for the many, what is that, cosmetic manufacturing industries. Okay, so many things is there: face cream, ointment, plaster, pencil, electric goods, and toothpaste, and so on. So, okay, media, and also used as a model, molding, making, and also printing industries, and also used the microtomy cancer cell section laboratory. Okay, the honeybee is also secreted. One of the gland is called as abdominal gland, otherwise called as abdominal wax gland. The abdominal wax gland is also called as cephalic gland and a resins. These both are present in it, collectively called as propolis. Propolis form the pollen grains. Pollen grain mostly present in the male reproductive part of the flower. Propolis is nothing but is a treatment used for the treatment of inflammation and the superficial burns. Bee venom is also one of the treatment used for the rheumatic arthritis in Japanese peoples in China and so on. So, bee diseases, nosema diseases, acrine diseases, aspergillosis diseases, and septicemia diseases. Fish production is nothing. So, what is that? Pisciculture. Okay, there are two types. Either one is called as capture fishing and also called culture fishery. Okay, where it will be, there are two things is there. One is called as sea water, river borne, and capture culture fishery is also things is there. Next will color of revolutions. There are different colors. There green revolution is denoted as a agriculture and study of agriculture. White revolution, milk and dairy. Black revolution, petroleum. Pink revolution, onion brands. Brown revolution, leather and cocoa. Blue revolution, fish. Gold revolution, horticulture. Yellow revolution, oil seeds. Likewise, other evolutions also given here. Yellow revolution, oil seeds. Golden revolution, fruits also there. Black and brown revolutions, non-convenience and energy. Silver revolution, egg. Don't forget, my dear, silver revolution's egg. Round revolution, potato. Last time I asked the questions. Red revolutions, tomato and meat. Gray revolution is also very, very important, mostly fertilizers. Okay, my dear. So remaining things we will see in the next class, my dear students. We will finish it up to this level only, my dear, today. Thank you. Take care. Bye. Thank you, ma'am.
Thank you, sir.
Thank you.